So I'm going to go ahead and get started in the interest of time. Welcome, everyone. We're super excited that you're all joining us today. Maybe some context for this event. So our team recently attended ABIM in Basel. While we were there, we heard a lot of buzz around microbial metabolites and their potential as, novel and potent active ingredients. At Ginkgo, we kind of believe and we have a strong hunch that production of these metabolites is going to require the use of tools like genetic engineering and other advanced biological tools. So what we wanted to do was create a forum for both R&D leaders and commercial leaders to learn about these technologies and some of the regulatory frameworks that influence their use. We have selected from the questions that you all submitted when you registered, but please feel free to ask more questions in the chat today or follow up with us directly to dive deeper into any of these topics. I'm going to get us started by passing it along to our host to introduce themselves.
Great. Yeah, I'll kick us off here. Hi, my name is Tammy Zimmer. I am a regulatory director at Compass. I've spent a majority of my career managing regulatory for biological crop inputs. I started my career at AgriQuest, which was subsequently acquired by Bayer Crop Science. So I spent 8 to 9 years in Bayer's federal regulatory team, after which I led regulatory efforts at Join Bio and Ginkgo Bioworks before joining Compass Regulatory. So happy to be here today.
That's me next. Hi, everyone. Thanks for joining. My name is Varghese Thomas. I'm the Senior Director for R&D programs at Ginkgo. I manage all the crop protection and crop nutrition portfolio programs for the various clients. Have about more than 15 years of experience in biological space, has a history of working in AgriQuest, then Bayer, and now Ginkgo. So I have touched the traditional biological products to now pushing using synthetic biology within various forms of programs to push for the next gen biologicals. So happy to be joining in this webinar. Looking forward to this discussion.
And hello, everyone. My name is Orpis Vasiliou. I am a senior microbial engineer at Ginkgo. I have worked a lot in the lab engineering microbes and also leading programs for the engineering of microbes. And I've been with Ginkgo for about 6.5 years. So how about we get started? Now, as we were on the 20th century, synthetic chemistry was the game. You wanted better fertilizers, you wanted something to control disease, you synthesize a molecule. And then as we moved forward, we had the traditional biologicals. You find a wild type strain that does something for you. Okay, it's good, maybe not as cheap, but maybe it's interesting. As we are now in the 21st century, what we need is next generation biological tools that can take us to a biological that is of sufficient quality and good price, so that we can have better efficacy and better cost. And Ginkgo has the biological tools to allow us to get there. And let's talk about 2 case studies to highlight that. In the first study, it's a little bit more close to traditional biologicals, but using very advanced tools. So traditionally, you would have a microbe, you'd mutagenize it, but when you're doing mutagenesis, it's a numbers game. How many mutants can you screen? 10, 20? How about 100? How about 1,000? How about many thousands? With Ginkgo's technology, we've got high throughput tools, or rather ultra high throughput tools, that allow us to screen thousands and tens of thousands and even hundreds of thousands of mutants to be able to find the mutant that has the ability that we are looking for. And in this specific case, we would do co-encapsulation of different mutants and pathogens. And after screening ultra high throughput and then looking a bit further into the top hits, we are able to find microbes that give us 4x improvement on a main primary active ingredient, 9x improvement on the secondary active ingredient. And that is just one case study. This thing has been repeated with many different microbes either for agricultural uses or other uses. Now, if you want to do something a little bit more bespoke, a little bit more advanced, you've got synthetic biology. And this is a case study actually where when we started the project, we had two options and we're pursuing two paths in parallel. One was with traditional mutagenesis and screening and making a customer strain better and better. But the other approach which we proposed was, all right, how about we take the genes that produce the molecule that the customer was interested in and express them in one of Ginkgo's hosts. And as you can see in the first expression, heterologous, we got a titer close to about a gram per liter. And then over 14 months, we're able to get it to have a 15x improvement from, let's call it, first drop. So through heterologous expression, iterative strain engineering, and step-by-step, we are reaching a titer that is commercially attractive to the client. We have actually verified it in our scale, transfer the strain to the customer, and they have verified it. And now we're pretty close to commercialization. We're just wrapping up some regulatory aspects. And speaking of regulatory and dealing with everything that is after or beyond the R&D, let me introduce you to Tammy. We've had a great experience working with her and with Compass, and she can take care of everything regulatory. Tammy, you're up.
Thanks so much. Appreciate that, Orpheus. Yeah, so Compass is well positioned here to help you navigate these regulatory frameworks for products derived from these technologies. Compass was founded with the goal of easing the regulatory burden for developers. We execute a variety of regulatory services with our team of experts. And what's unique about us is that we also have a software platform that helps supports us. Our software database helps us track and manage regulatory activities to help bring increased efficiency, transparency on the status of authorizations, and ultimately improving compliance. We have in-house experts which span all product types, and then specialists like myself working in this new and emerging technology area. We provide regulatory support really across all stages of development, from early research through to commercialization, defining regulatory requirements and securing those authorizations that are needed for your R&D activities, making sure you incorporate those into your development timelines. We can facilitate early engagement with regulators, develop data strategies, manage your studies, and really drive your dossier development and submission. And then we also help track and manage your existing authorizations, handling any amendments, label updates, as well as renewals and sales reporting at the state level, which can be really quite tedious here in the US, and that's where our software really helps us out. As I mentioned, Compass does have expertise navigating pathways for emerging technologies. So I'm really excited to be here today to have this conversation and to talk about the different paths that are available for these technologies. So thank you.
All right, thank you for the intros. So I'm going to start us off with the set of questions that we got from attendees asking about definitions that regulatory bodies use. So I think, Tammy, that's in your wheelhouse. We'll start there.
Yeah, definitely. So to before we jump in, I think let's start with the different types of products and just talking about the types of products that can be generated from GE microbes. So your product could result in the inactivated or killed genetically engineered microbe being present in the final product, yet that still contains your non-viable GE microbe, as well as all the metabolites and proteins that may be of interest for your product. You could develop a cell-free lysate of a genetically engineered microbe, right? So you still have those metabolites and proteins of interest, but you don't have the whole cells. You do have the residual DNA of the genetically modified cells. Or you can make a purified biochemical where you're isolating that protein or metabolite of interest, and there are no cells or residual DNA that's present in that final product. So really, depending on the active substance that's being produced in your intended market, there are different regulatory approaches that can be used to fit your product into a biological specific pathway, whether it's, you have a microbial metabolite in your end use product, and you are the inactivated, killed whole GE microbe cells are still present. You could pursue a microbial pesticide path depending on your geography. If you have a metabolite that is in a cell-free or a purified biochemical metabolite substance, then you could potentially pursue a biochemical path in those geographies. I know that one of the questions was about de novo protein, with a novel function. If you have a de novo protein with a novel function and there's no sequence homology to a natural protein, you would face higher regulatory burden due to its unknown safety profile. However, if you have a natural protein or even an engineered version of a natural protein, you may potentially be able to register it as a biochemical, depending on, again, your geography. So we do have the US and Brazil have specific biochemical definitions and pathways for these types of products. So if you have active substance that meet the key criteria, which is in the US and Brazil, the substance needs to be naturally occurring or synthetically identical and have a non-toxic mode of action. I do want to point out that the technical definition under the new bioinputs law in Brazil, which we'll talk about in a little bit, is not yet defined. So something to keep an eye out to see how that may change. In the EU, biochemicals are regulated as chemical active substances, but they may qualify as a low-risk active substance and get streamlined treatment if they pose no hazards. So if your active substance really fits these criteria for the intended market, the regulatory data requirements and review times will be much faster than that of a conventional chemical. And even if you have a biochemical which let's say it doesn't meet this non-toxic mode of action, for example, if it still has a low toxicity profile, you're still able to negotiate a reduced data package. So there are avenues. Next slide.
I think there's a third question, Tammy, around, what is the working definition of genetically engineered biologicals? How would you address that?
I would say I would love to have a working definition of genetically engineered biologicals. There's no harmonized regulatory definition for genetically engineered biologicals, not even across the agencies within the US and the coordinated framework, right? APHIS-BRS has a regulatory definition of genetic engineering, but EPA under either TSCA or FRISCA or FIFRA does not. So each agency has their own regulatory definitions that will trigger jurisdiction under that agency. So for example, under TSCA, only intergeneric microorganisms are regulated, and those, which basically means those developed with DNA from different genera. So it's really critical to engage your regulatory experts from the start to understand what those regulatory triggers are and identify any jurisdiction and authorizations that are needed for you to conduct your research and development activities. And then don't forget to incorporate those timelines into your development timelines and your development plan. So for example, if you're developing a purified biochemical, that product type that we showed, even though the GE microbe is not in the final product, it still may be subject to BRS or TSCA here in the US or CTN Bio in Brazil. So you really need to understand the regulatory considerations there. So bring regulatory in from the very beginning. That'll be key. So it's also, you can also think about strategies to try to mitigate some of these triggers if it works for your technology. It doesn't always fit for everything, but there are strategies you can employ to try to reduce these regulatory requirements from host organism or gene source selection, things of that nature. I also want to emphasize that frameworks and implementations of regulations evolve over time. So regulators, their interpretation and implementation of the regulations may differ from yours. You can read a regulation and think, oh, this makes sense. And then you're like, wait, what? So it's really critical to engage early with regulators and just to introduce your technology and align on a regulatory path. It's important to confirm your jurisdiction or non-jurisdiction just to ensure your freedom to operate. So you obtain the proper authorizations for your R&D and commercial activities. It's also critical to identify any potential regulatory concerns early, especially if you're developing a novel technology. You really want to get just that initial feedback from the agency to see if there are any concerns raised and how you may be able to mitigate those strategies and incorporate them into your development process.
Amy, so I think this next set of questions that we got were, they also fall into your wheelhouse. We had a number of questions around regulatory frameworks in key geographies. Can you speak to some of the differences across regions?
Yeah, happy to. We are seeing some shifts in the three key markets here that we're talking about. One of the most exciting is that Brazil has recently implemented a new bioinputs law, which establishes a dedicated framework for biological products. This is really promising to accelerate market entry for these types of products in Brazil. MAPA is still the lead agency for registering these products, and Visa and Ibama would only come in and be involved for new pesticide products, essentially. CTN Bio is still responsible for assessing the biosafety of any genetically engineered microorganisms that would fall under their jurisdiction. The new law excludes biological products from the existing pesticide and fertilizer laws. As I said, it establishes a dedicated framework. And the law really broadly defines bioinputs, so it's including biotech-derived products as well as those that are structurally similar and functionally identical to natural ones. The law was published at the end of last year. However, the regulations implementing the law are still in development. When the law was published, there was a decree. They had to issue the regulations, I believe, within 360 days. So keep an eye out on how that's evolving there, and it will be really exciting to see how those get implemented and how it improves the process in Brazil for bioinputs. The US has an established framework for the regulation of biotechnology products, and it's a coordinated framework with the three primary agencies being EPA, USDA, and FDA, and there have been continuous efforts to modernize that framework through the past administrations. About probably 7 years ago now, EPA, their Biologics Pollution Prevention Division, which registers all of the biological products, restructured to form the Emerging Technologies Branch. In 2020, they implemented the APHIS BRS new secure rule. However, in 24, that rule was vacated and the agency has to revert to the previous regulations. So there's still some evolving frameworks there for GE microbes or GE biostimulants and biofertilizer products that are live. organisms. I know there was another question there regarding that. You definitely need to watch the evolving framework in the US for those products. There are some commercial challenges under the existing framework if your product is regulated under BRS. But as we discussed earlier, there are mitigation strategies you can employ to see if you're able to develop organisms that fall outside their jurisdiction. In the EU, biologics are still evaluated under a chemical framework. However, new microbial rules have been implemented to provide clarity and streamlined approvals. There's still no dedicated path. Short-term efforts are being made to make improvements under the existing framework, but long-term, the goal is for of both the European Parliament and industry is to establish a dedicated framework. But I think that's going to take a bit. So, thanks.
All right, thank you, Tammy. So this next set of questions, we saw a theme amongst the questions here around, where are biologicals going? What are the kind of new technologies that are going to be employed? What is kind of the burden of proof to ensure that growers will adopt these biologicals? So Varghese, you've been in biologicals for decades now. We'd love to have you speak to these.
Yeah, thank you. So really good questions. One of the things that I am excited about working currently now is that there's more adoption of biologicals. It's becoming more like a major pillar among similar to what now where chemistry is or traits are or seeds are. So it's pretty good. Like now we don't have to do a lot of advertisement for biologicals. The growers are accepting that biological is part of the ag system now. So it's really good. What's happening now is that still biologicals are not perfect, like the traditional biologicals that we've been working for last few decades. Now there's new generation of biological that's coming that we have seen. Also, we saw that in ABIM is that more metabolite enriched products as well as more biomolecule enriched products like proteins, peptides, or RNAi, and then also for pheromone based. So there's many of those nature-inspired products that's now coming, not limited to the live micro product that used to be the case for last few decades, let's say. So pretty exciting place to be. That goes towards the number 2 question, where what is the vision of the future? Is the adoption of the next generation technology a differentiation strategy? Partly, yes, it's a differentiation strategy, different from being part of the traditional ag biological of the live microbe, but more of also addressing the challenges that the growers have faced with the traditional products, which was more of lack of consistency, lack of performance. So now with these more metabolite enriched products and also biomolecule enriched products, what we are seeing is much more closer, better performance, which is more closer or can become par. Not yet, but at least we believe that with the results that we are seeing, the performance will be at par with the small molecule chemistry. And that's the ultimate target that all these companies are striving towards. Will one solution be the silver bullet? No, I believe it would be a combination of all these tools coming together. So that's good that we have more diversity of these tools coming within the biological framework and not limited to that one first used to be the live micro product concept. So that kind of like these are the key areas that now we are seeing other than the live microbe or the microbial based. It's RNAi. There's a lot of clients that now we are working with who are interested in the gene edited microbes as well as going towards more biomolecule enriched products. Where Ginkgo, what we've been working with is more, we have those tools where we can, similar to what Orpheus was talking about, for non-GM, we have NCAPs or the mutant-based technologies, which can be done very high throughput. For GM, it's much more straightforward. We have high throughput ways to engineer wild-type microbes, as well as we have production host strains for things which are more friendly to be expressed under a chassis. So, and we can do also the fermentation optimization as well as go up to the scale up for some of the key new products that's coming into the market. So with that, looking at the time, I would pause. I hope that this was a good kind of like a topic to come together where the market is going. If you go to the next slide, Dia, that shows that what are the different frameworks where Ginkgo has been working, because that also tells us where the market is driving. So we have this active metabolite-based discovery as well as mode of action-based discovery that we do, which we have been working with the clients, as well as we focus on the fermentation formulation as well as the engineering tools. So there's all these tools that come together that enable us to build a better solution, which could be the next generation solution that can enable better performance in the field.
Great. Well, thank you to all of our speakers and our attendees for submitting questions and answering questions. If you have more questions, please reach out. We will send through a form where you can sign up for, free consultation with Compass or Ginkgo to learn more about any of these topics. And appreciate your engagement. And any questions that we're not able to